Introduction to Pharmacoepidemiology Part 2. Learning Objectives. Understand drug approval process. Identify importance and role of animal studies. Understand the limitations of preclinical studies. Drug approval process. The first steps in the development of a new drug are the discovery or synthesis of a potential new drug molecule and seeking an understanding of its interaction with the appropriate biological targets. In addition to in vitro studies, relevant biological effects, drug metabolism, and pharmacokinetic profiles, and particularly assessment of the relative safety of the drug must be characterized in animal studies before human testing. With regulatory approval, human testing can go forward in 3 phases before the drug can be considered for approval for general use. A 4th phase of data gathering and safety monitoring is becoming increasingly important and follows after approval for general use. As you can see in this figure, this shows the time frame for development and testing process required to bring drug to the market. So as you can see, the process takes about 20 years in total. So we start with in vitro studies, biological products and chemical synthesis to identify the lead compound. After this takes about 2 years, then we move forward for animal testing to detect the efficacy and selective mechanisms. After results obtained from the animal testing, this applies to the drug approval process in the USA. So investigational new drug is applied. And then human studies starts, clinical trials. So 3 phases. Phase 1, detecting the pharmacokinetic profile. Phase 2, detect the efficacy. Does it work in patients? Phase 3, does it work in double-blind design? We will discuss this later. All through clinical testing, we assess drug metabolism and safety assessment. This takes about 5 to 6 years. So in total, 8 to 9 years, a new drug approval application is applied. And then we move to phase 4 study, post-marketing surveillance. As you can see, the patency of any new drug expires after 20 years after filling of application of the investigational new drug. Generic become available after that. This is very important figure to consider. So again, to summarize, we start with biological data, structure level, in vitro, animal testing, and we go through phase 1, phase 2, phase 3 studies until the drug is approved by the FDA and released to the market. Drug discovery. Most new drugs or drug products are discovered or developed through one or more of 6 approaches. Identification or elucidation of a new drug targets, rational drug design of a new drug based on understanding of biological mechanisms, drug receptor structure and drug structure, chemical modification of a known molecule, screening for biological activity of large numbers of natural products, banks of previously discovered chemical entities, and large libraries of peptides, nucleic acids, and other organic molecules. Biotechnology and cloning using genes to produce peptides and proteins. This is a very hot field and new field in drug discovery. So efforts continue to focus on the discovery of new targets and approaches, from studies with genome, proteomics, nucleic acids, and molecular pharmacology for drug therapy. And finally, combination of known drugs to obtain additive or synergistic effect or repositioning of a known drug for a new therapeutic use, finding new indications for new drugs. Preclinical testing. So in preclinical testing, we have different experimental methods or target organs We can use also species or tissues to measure certain outcomes. So for example, we can have molecular level and we can have cellular level. In cellular level, we can use cultured cells or isolated tissues to study, for example, the effect, for example, isolated tissue, blood vessels of the heart, to study the effect on vascular contractions and relaxation, selectivity for vascular receptors, effects on other smooth muscles. If we go to molecular level, we can use study cytochrome P45, target tissue is the liver, to see enzyme inhibition, effect on drug metabolism, for example. These are all areas we assess in preclinical safety and toxicity testing. The most essential part of the new drug development process and why we conduct the animal studies is to assess correctly the limiting toxicity of a drug and the therapeutic index comparing benefits and risk of a new drug. This table shows types of safety measures that we assess in preclinical studies, in animal studies. You can see that we have different types of toxicities measured: acute toxicities, subacute, chronic, carcinogenic, effects on reproductive performance, mutagenic, investigative toxicology. Let's take a mutagenic example. So in animal studies, we need to assess the effect on genetic stability and mutations in bacterium or mammalian cell in culture, dominant lethal test and clastogenicity in mice, for example. Also, subacute toxicity, for example, 3 doses, 2 species. We take 2 species, 2 different species. 4 weeks to 3 months may be necessary prior to clinical trial. The longer the duration of expected clinical use, the longer the subacute test can be detected. All these are measured in preclinical data. So also we measure the dose, the lethal dose or the toxic dose. So we have 3 types of doses. We have no effect dose, which is the maximum dose at which a specified toxic effect is not seen. The minimum lethal dose, which is the smallest dose that is observed to kill any experimental animal. The median lethal dose, which is the dose that kills approximately 50% of the animals. These doses are used to calculate the initial dose to be tried in humans. So we extrapolate the results of preclinical data to obtain initial dose that we can start with the clinical data on humans. Limitations of preclinical testing. Toxicity testing is time-consuming and expensive. 2 to 6 years may be required to collect and analyze data on toxicity and estimates of therapeutic index before the drug can be considered ready for testing in humans. Large numbers of animals may be needed to obtain valid preclinical data. Scientists are properly concerned about this situation, and progress has been made toward reducing the number required while still obtaining valid data. Cell and tissue cultures in vitro methods are increasingly being used, but their predictive value is severely limited. Extrapolation of therapeutic index and toxicity data from animals to humans are reasonably predictive for many, but not for all toxicity. So we can't obtain sufficient data for all toxicity from extrapolation of animal studies. We still can find new findings in human studies, which is risky. Finally, for statistical reasons, rare adverse effects are unlikely to be detected because the number of animals used in animal studies is considered low in general to attain or to observe rare adverse effects.